The techniques used in CRISPR. CRISPR, clustered regularly interspaced short palindromic repeats, is a revolutionary genetic engineering technique that involves several key steps. Adaptation. DNA from an invading virus is processed into short segments and inserted into the CRISPR sequence as new spacers. Production of CRISPR RNA. The CRISPR repeats and spacers in the bacterial DNA undergo transcription, which is the process of copying DNA into RNA, ribonucleic acid. Targeting. CRISPR RNAs guide the bacterial molecular machinery to destroy the viral material. In the lab, scientists use CRISPR/Cas9, where Cas9 is an enzyme that acts like molecular scissors to cut DNA at a specific location. This system can be directed to disable or repair genes, or insert new sequences. Newer versions, called base editors, can edit the genetic material one base at a time without cutting, acting more like a pencil than scissors. CRISPR technology has been used to fix genetic diseases in animals, combat viruses, and even prepare pig organs for human transplants. It's a low-cost gene editor that's easy to use, making it possible for researchers to explore the basic mysteries of life in ways that were previously difficult or impossible. The technical processes using CRISPR. CRISPR technology is a sophisticated method of gene editing that allows scientists to modify an organism's DNA with precision. Here's a technical breakdown of the process using CRISPR-Cas9, the most commonly used system design of guide RNA, gRNA. The first step is to design a gRNA that matches the DNA sequence of the target gene. This gRNA will direct the Cas9 enzyme to the exact location in the genome where an edit is desired. Formation of the gRNA, Cas9 complex. The gRNA is combined with the Cas9 enzyme, forming a complex. This complex will navigate through the cell's nucleus to the DNA targeting. Once inside the nucleus, the gRNA, Cas9 complex locates the target DNA sequence by matching the gRNA with its complementary DNA sequence, DNA cleavage. When the target site is found, the Cas9 enzyme acts as molecular scissors and makes a precise cut in the DNA strand at the location complementary to the gRNA. DNA repair and gene editing. After the DNA is cut, the cell's natural repair mechanisms kick in. Scientists can utilize this repair process to introduce changes to the genome. There are two main pathways for repair: non-homologous end joining, NHEJ. This pathway often results in insertions or deletions, indels, at the cut site, which can disrupt the gene function. Homology-directed repair, HDR. If a DNA template with the desired sequence is provided, the cell can use it as a blueprint to make a precise edit, such as correcting a mutation or inserting a new gene. Verification of editing. After the CRISPR-Cas9 system has done its work, scientists check the edited cells to confirm that the right changes have been made to the DNA. This process has been used for various applications, from basic research to potential treatments for genetic diseases. It's a powerful tool because of its simplicity, efficiency, and versatility in editing genetic material. Scientists have developed several methods to deliver CRISPR components into cells, each with its own advantages and applications. Here are the primary methods. Electroporation. This technique uses a short pulse of electrical current to temporarily increase cell permeability, allowing the CRISPR components to enter the cell. Liquefaction. In this method, lipid molecules form a bubble-like structure called a liposome to encapsulate the CRISPR components, which then fuse with the cell membrane to deliver the contents into the cell. Microinjection. This is a direct method where CRISPR components are injected into cells or embryos using a fine needle. Nanoparticles. Nanoparticles can be designed to carry CRISPR components and deliver them into cells by merging with the cell membrane. Viral vectors. Modified viruses can be used to deliver the genetic material required for CRISPR into cells. Common viral vectors include lentivirus, adenovirus, and adeno-associated virus, AAV. Each of these methods can be used to deliver the necessary CRISPR components, such as the Cas enzyme and guide RNA, into a variety of cell types for genome editing purposes. The technical aspects of electroporation in CRISPR. Electroporation in CRISPR technology is a method used to introduce the CRISPR/Cas9 system into cells, particularly zygotes, for genome editing purposes. Here are some technical aspects of electroporation in CRISPR. Electroporation mechanism. It involves applying an electrical field to cells to temporarily create pores in the cell membrane, allowing the CRISPR/Cas9 components to enter the cell. CRISPR/Cas9 components. synthetic CRISPR/Cas9 components, which include guide RNA and Cas9 protein, are delivered into the cells through these pores. Optimization for efficiency. Researchers have optimized electroporation protocols to improve the efficiency of CRISPR/Cas9 genome editing. This includes adjusting the electrical parameters to minimize cell damage and maximize uptake of the CRISPR components. Advantages over pronuclear injection. Electroporation is considered a rapid alternative to pronuclear injection, which is a more elaborate procedure. It can lead to less physical damage to the zygotes, resulting in improved embryo development and potentially higher rates of successful genome editing. EASY method. A novel approach called easy electroporation of zygotes, EASY, allows for the efficient generation of transgenic mice using a common electroporator without the need for specialized equipment or corrosive pretreatment of zygotes. The technical aspects of liposuction in CRISPR. Liposuction is a technique used in CRISPR to introduce genetic material into cells. Here are some technical aspects of liposuction in CRISPR. Definition. Liposuction is a form of liposome-mediated transfection that uses lipophilic reagents to facilitate the cellular uptake of nucleic acids, such as the CRISPR/Cas9 system mechanism. It involves encapsulating the CRISPR/Cas9 components within liposomes, which are then able to merge with the cell membrane and deliver the genetic material into the cell via endocytosis. Optimization. Studies have focused on optimizing the timing and duration of liposuction to improve gene editing efficiency. For instance, in porcine zygotes, adjusting the liposuction parameters affected the rate of successful gene edits. Advantages. Liposuction can be a simpler alternative to other methods like microinjection or electroporation, as it doesn't require sophisticated equipment or techniques, making it accessible for more laboratories. Challenges. Despite its simplicity, achieving high gene editing efficiency with liposuction can be challenging, and ongoing research aims to refine the process to increase its effectiveness. The technical aspects of microinjection in CRISPR. Microinjection in CRISPR is a precise method used to introduce genetic material directly into cells, including embryos, for genome editing. Here are some technical aspects of microinjection in CRISPR mechanism. It involves using a fine glass micropipette to inject the CRISPR/Cas9 components, such as guide RNA, gRNA, and Cas9 protein or mRNA, directly into the nucleus or cytoplasm of target cells. Precision. Microinjection allows for the delivery of precise quantities of the CRISPR/Cas9 components, which is crucial for achieving targeted gene editing without affecting other parts of the genome. Application. This technique is widely used in various organisms, from plants and animals to human cells, and is particularly important in developmental biology and genetics research. Challenges. Microinjection requires skilled technicians and specialized equipment. It can be time-consuming and has a risk of damaging the cells if not done carefully. Advancements. Research continues to improve microinjection methods for different species, such as crustaceans and plants, to enhance the efficiency and success rates of CRISPR/Cas9 genome editing. The technical aspects of nanoparticles in CRISPR. Nanoparticles offer a novel approach to deliver the CRISPR/Cas9 system into cells for genome editing. Here are some technical aspects of using nanoparticles in CRISPR delivery efficiency. Nanoparticles can enhance the delivery efficiency of CRISPR components into cells, which is crucial for successful genome editing. Non-viral carriers. They serve as non-viral carriers, providing a safer alternative to viral vectors, which can integrate into the host genome and cause unintended effects. Customization. Nanoparticles can be tailored in size, surface charge, and functionality to improve cellular uptake and target specific cell types or tissues. Release mechanism. Some nanoparticles are designed with disulfide bonds that break down inside the cell, releasing the CRISPR components efficiently. Extracellular vesicles. Research is exploring the use of extracellular vesicles, a type of nanoparticle, for CRISPR delivery, which could mimic natural cellular processes. The technical aspects of viral vectors. In CRISPR, viral vectors are a key tool in CRISPR technology, used to deliver the CRISPR/Cas9 system into cells for genome editing. Here are some technical aspects of viral vectors in CRISPR delivery mechanism. Viral vectors transport the CRISPR components, guide RNA and Cas9 protein, into the host cell by exploiting the natural ability of viruses to infect cells. Types of viral vectors. Commonly used viral vectors include adenoviral vectors, AD versus adeno-associated viruses, AAVs, and lentivirus vectors, LVs, each with its own advantages and limitations. Efficiency. Viral vectors are preferred for their high delivery effectiveness, which is crucial for successful genome editing applications. Safety concerns. While viral vectors are efficient, there are concerns about their potential to integrate into the host genome, which could lead to unintended consequences. Advancements. Research is ongoing to improve viral vectors for CRISPR delivery, focusing on enhancing safety, specificity, and efficiency. How base editing works. Base editing is a refined form of gene editing that allows for the direct conversion of one DNA base into another, without the need for double-stranded DNA breaks, DSBs, typically associated with CRISPR/Cas9. Here's how it works. Design of base editors. Base editors are engineered proteins composed of a modified CRISPR/Cas9 system fused to a deaminase enzyme. The Cas9 component is typically mutated to act as a nickase. It can only cut one strand of the DNA, not both, targeting the DNA. Similar to CRISPR/Cas9, a guide RNA, gRNA, directs the base editor to the target site in the DNA sequence where the edit is needed. Chemical conversion. Once the base editor is at the target site, the deaminase enzyme chemically alters the target DNA base. For example, a cytosine, C, can be converted into a thymine, T, or an adenine, A, into a guanine, G. DNA repair. The cell's natural DNA repair processes then solidify the change, resulting in a permanent edit. Base editing is particularly useful for correcting point mutations, changes in a single base pair in DNA, which are a common cause of genetic disorders. It offers a more precise editing option with reduced risks of unintended edits elsewhere in the genome, making it a powerful tool for research and potential therapeutic applications.